Assalamualaikum and hi everyone, I am Miss Siti Azman, and in this video I will be explaining to you about anaerobic respiration. At the end of this topic, you are expected to understand about anaerobic respiration, the difference between aerobic respirations, and also able to explain about alcohol fermentation and lactic acid fermentation, which are part of anaerobic respiration reaction. Before we talk about anaerobic respiration, have you ever wondered how yeast, an organism, can turn barley malt into beer, and also how your muscles can work even though they are low in oxygen? So these are all due to these alternative pathways to break down food molecules. We call that as anaerobic respiration and fermentation. So these alternative pathways break down glucose in order to generate ATP without using aerobic respirations. From the previous videos that you have covered, we know that the main process to generate ATP is through aerobic respiration, where oxygen is being used. However, there are certain situations where aerobic respiration is not possible. Aerobic respiration is not possible when oxygen level is low or oxygen is not around. So why oxygen is important? Because oxygen acts as an acceptor at the end of electron transport chain. Let's just recap a little bit about electron transport chain. We know that in aerobic respiration, the majority of ATP is being produced through oxidative phosphorylation, where electron from NADH and FADH2 molecule will be transferred to these complexes, and at the end we have chemiosmosis where ATP will be generated. If you look at the end of electron transport chain, we have oxygen molecule that act as a final electron acceptor. If there is no oxygen, electron transport chain will stop. So the whole process will stop because there is no molecule to accept this electron. When this happens, certain organisms can switch to alternative pathways, pathways to generate ATP even without the presence of oxygen. So the alternative pathways to generate ATP without oxygen, we call that as anaerobic respiration. So the reactions in anaerobic respirations involve glycolysis plus fermentation, or anaerobic respiration could also happen through glycolysis plus anaerobic respiration. So we will see a little bit why we call that as anaerobic respiration. And anaerobic respiration only happens in cytosol. So it's not going to happen in mitochondria. Before we go further about anaerobic respirations, let's look at some of anaerobic organisms. Anaerobic organisms can be divided into two types. We call them as facultative anaerobes. When we talk about facultative anaerobes, it refers to organisms that can switch between aerobic and anaerobic respirations. When oxygen is present, they will undergo aerobic respiration to produce ATP. When the oxygen level is low, they will switch to anaerobic respiration. The other type of organisms, we have obligate anaerobes. Obligate anaerobes refer to organisms that only depends on anaerobic respirations in order to generate ATP. To a certain extent, this organism will die when oxygen is present. So they totally did not use aerobic respiration. So the mechanisms to generate ATP without oxygen, or we call it as anaerobic respiration, can be divided into two mechanisms. The first one is anaerobic respiration with electron transport chain. But this is different than aerobic respiration because their final electron is not oxygen. Okay, so they did not use oxygen as their final electron acceptor. Instead, they use other molecules such as sulfate, nitrate, and also sulfur. The other mechanism to generate ATP without oxygen, we have fermentation. Okay, so fermentation can be divided into two types: alcohol fermentation and also lactic acid fermentation. When we discuss anaerobic respiration, we are going to focus more on fermentation process. Before we talk about fermentation, okay, let's just recap about cellular respiration, okay, the one that you have covered in previous videos. Okay, cellular respirations or aerobic respiration involve the process of glycolysis, pyruvate oxidation, Krebs cycles, and at the end we have electron transport chain. Most ATP is being generated here. How they are being generated? The NADH molecule and FADH2 molecule, okay, the electron carrier from glycolysis, from pyruvate oxidation, from Krebs cycle will be brought to electron transport chain, okay, and then the electron will be transferred, and at the end of it we have chemiosmosis, okay, where ATP is being generated. Once the electron carrier is being brought to electron transport chain, okay, for example NADH, okay, so they will undergo oxidation, okay, where they lose electron, okay, because the electron has already entered electron transport chain, and NADH now becomes NAD plus molecule. Okay, so all NADH from glycolysis, pyruvate oxidation, Krebs cycle, once they reach electron transport chain, they undergo oxidation and they become NAD plus. So what is the purpose of NAD plus? These NAD plus molecule will be recycled in glycolysis, in pyruvate oxidation, in Krebs cycle. However, in fermentation, we know that fermentation is the generation of ATP without oxygen. So there will be no pyruvate oxidation, no Krebs cycle, no electron transport chain. So when there is no electron transport chain, NADH cannot be oxidized into NAD+. What happens is you will have accumulation of NADH. If you have accumulation of NADH, there is no electron transport chain, ATP cannot be generated. So what happens in fermentation is we need a certain reaction after glycolysis that will regenerate NAD plus from NADH. So to look closely why NAD plus is important, why do we need to regenerate NAD plus? We have to go back to glycolysis reaction. Imagine this is the environment where oxygen is low. So when oxygen is low, there will be no Krebs cycle, no pyruvate oxidations, no electron transport chain. You will have accumulation of NADH. Okay? And after a while, the amount of NAD plus will decrease in the cell, and later on there will be no NAD plus. If there is no NAD plus, this particular step cannot happen. Where glycerol 3-phosphate will be oxidized, NAD plus will be reduced, and if that step doesn't happen, the consequent step will not going to happen as well. Okay, there will be no next steps as well. So there will be no ATP being generated. And the amount of ATP in the cells will get decreased later on, and it will get decreased more and more. So that is why it is important to regenerate the NAD+, so that when there is NAD+, this reaction can proceed. The ATP can be produced through substrate level phosphorylation. this step and this step at the end. I hope you understand about anaerobic respiration and about fermentation, okay, and why is it important to regenerate the electron carrier NAD+. In the second part of anaerobic respiration, I will be explaining about the two common types of fermentation reaction. Okay, thanks for watching. Bye.